Thank you so much, Regina, for the introduction. I am very excited to be here to present about our work in Alzheimer's disease. And before starting the presentation, I wish to thank J Clinic for their support. That was very critical in the early stages of this work. Having their support, we were able to move ahead with this project. It was actually a project that was funded last year, and I'm happy to share that we now have NIH funding to make much more progress in this work on Alzheimer's disease. My name is Dimitrios Pantazis. I'm principal research scientist at MIT. And this work is actually a joint effort by a large group of investigators, including Munjia Xu, a postdoc in the lab that has done a lot of work that I will show here, Changjing Li, associate professor in Harvard Health Science, and other collaborators. Fernando Maestro, very critical work. He has been working decades in Alzheimer's disease in Madrid, and many others like John Moser, Michael Funk, and others in University of Texas Health Science Center. So once I found the way to move to the next slide, okay. So it is difficult to overstate the importance of Alzheimer's disease. It is the most common form of dementia with very debilitating symptoms. It's a devastating disease. Symptoms include memory loss, cognitive dysfunction, anxiety, aggression, and more. Back in 2019, it was affecting 41 million people, for example, and this is scheduled to more than triple. It's planned. I'm sorry. It will more than triple in the 2050s unless we do something about it. 1 in 6 women and 1 in 10 men who live past the age of 55 will develop dementia. And the healthcare cost is enormous, projected to be 1.1 trillion in 2050. Before proceeding further, I do want to mention also that in this seminar series, we aim to also have a clinical collaborator here to talk about Alzheimer's disease and, like all this relevant work here. Unfortunately, I will not have one here because the majority of my team is actually far remote in Texas or in Madrid. An AI expert here, Changjing Li, actually has a milestone meeting with a private company, something that you cannot really postpone. So I will be alone this time. So I will try to make justice, of the clinical part as well, though by far I'm not an expert in this, but I hope to give you a complete view of this work. So closing this parenthesis, acknowledging the importance of the disease, the National Institute of Health and the Alzheimer's Association have been pushing ahead for more funding in this direction. And funding in Alzheimer's disease has increased 7-fold since 2011-12. So it's an enormous amount of funding directed in this research, $3.2 billion this year. And this is because these institutes acknowledge the need for an early detection of Alzheimer's disease, to make progress in this direction, to understand the pathology, and then eventually to develop drugs. And with an early detection, we can also work on patient management, behavioral intervention, many other approaches and research proposals supported by these institutes. So Alzheimer's disease is actually a very complex disease. It has complex etiology and it has multiple pathogenesis. And there is not a very clear understanding in the community about all these mechanisms. There's vast research on these directions. But what we know well is that the hallmarks of Alzheimer's disease are the accumulation of two types of proteins in the brain over decades. One is amyloid beta, and I think you can see my cursor, I hope. So amyloid beta is one protein, tau is a second protein that accumulate over a long time in the brain. And actually you can see that amyloid beta starts 20 years or more before the clinical manifestation of the disease. When we have...
It starts 20 years.
So this is by studying the accumulation of protein at different ages and knowing the average age that have Alzheimer's disease. And actually this should be longitudinal studies of very long time now. I think the first case of Alzheimer's disease, like reported by Alzheimer, Alois Alzheimer, was in the early 90s. And our knowledge has increased a lot over the years, especially in the recent times. But also to say that these are kind of, hand wavy curves, because it really, the trajectory varies a lot per individual. And in some patients, it goes really fast. In others, especially elderly patients, it's a much more slow progression, and that lasts many, many years. That was a good question, though. At later stages, we do have changes in brain structure like atrophy and, of course, cognition, which leads to the clinical diagnosis. Amyloid beta and tau accumulate in different forms. So amyloid form plaques outside the cells, whereas tau forms neurofibrillary tangles inside the cells. The mechanisms on how they work and interact with one another are not known, or there are many, many hypotheses and theories about it. But it's generally accepted that are toxic to the synapses of the neurons, and they also need to lead to neuronal death. And it's reasonable to anticipate with synapse pruning and neural death. We also have functional connectivity alterations and structural connectivity alterations in the brain that also span a long time. And it's actually the focus of this work that I'm presenting here, to use these connectivity alterations as biomarkers of the disease, rather than this accumulation of proteins, which are very difficult and costly to measure. In terms of prediction and diagnosis of AD, the first set of clinical criteria were focused on symptoms and neuropsychological tests. So we have tests such as the clinical dementia rating, the mini mental state examination, and the Montreal Cognitive Assessment, or MoCA score. These are tests showing a cognitive decline. But in more recent efforts, the community focused on a definition of criteria that removes from the syndromal construct and goes more to a biological construct, which is better defined. So we don't rely on clinical consequences of the disease and let me stress that, in a research framework, not in a clinical framework, but in a research framework. We don't rely on the symptoms of the disease, but rather on in vivo biomarkers now, because they have become available and they're very reliable and they lead to objective definition of the disease. So we have in vivo biomarkers of amyloid beta, of tau, and of neurodegeneration, or with capitals A, T, N. And this is the research framework that we use, the recent research framework that we use now, where A or T or N are denoted with plus or minus, depending on whether we have manifestation of the disease symptom of the disease biomarker. So we can have A plus, T minus, N minus, and so on and so forth. And the amyloid beta biomarker defines that someone is in the Alzheimer's spectrum, whereas the T, the tau, defines if someone is actually in Alzheimer's disease. And then N, for neurodegeneration, quantifies the staging severity. So these are the biomarkers used today. And let me show you some examples. FTG PET is a biomarker of metabolism in the brain, so measures brain function. And in the brain of Alzheimer's, of a patient with Alzheimer's disease, an elderly patient, you can see in this scan hypometabolism in parietal areas denoted with the arrows, whereas the normal scan has more uniform uptake throughout the cortex. Another biomarker is structural MRI. Probably all are familiar with this in this audience. So in this case, we see atrophy in the Alzheimer's disease patient, more or less throughout the cortex, actually. You can see, for example, in ventricles or in the hippocampus, the devastating results of the disease. And another biomarker is amyloid PET. PET is for positron emission tomography. And in this case, we also see a strong uptake of the amyloid PET throughout the cortex, showing that there is a large accumulation of amyloid in this patient. And last, the most recent of these is tau PET in recent years, and it shows, it uses tracers for PET protein, and it shows in this patient the uptake of tracer in entorhinal cortex, hippocampi, parahippocampal regions, and temporal and occipital lobes, all marked with the arrows. So these are very valuable biomarkers, but they are difficult to acquire. And that's because so PET and MRI are actually expensive, and PET also involves radioactivity. Another biomarker is CSF and cerebrospinal fluid, and that can be collected with a lumbar puncture, but many patients would find this objectable. And all of these need to be done in a hospital setting. So it's clear that these biomarkers cannot be used to scan the broad population, definitely not in home. So we need more affordable biomarkers that can be done with a low cost in home or even in a residential setting. Otherwise, as it is done now, Alzheimer's disease is diagnosed late, usually with when the symptoms are very pronounced, and then we collect these biomarkers. By that time, we have irreversible neurodegeneration, and it's too late to intervene. So in this work, we focused on technologies that can capture brain function, which could be a surrogate of the synapse degeneration and functional alterations in the human brain caused by Alzheimer's disease. Of these popular technologies, the left ones have to do with measuring hemodynamics, capturing hemodynamics. So basically when neurons are active, they consume oxygen, and the body provides more oxygenated hemoglobin. So these modalities measure the ratio of oxy versus deoxygenated hemoglobin. One of them is functional MRI, the other is near-infrared spectroscopy. Functional MRI would be too costly to conduct in an ambulatory setting. It's not feasible. And near-infrared spectroscopy cannot see deep inside the brain. So there are obstacles in these methods. Here, I will focus on MEG and EEG. MEG measures the magnetic fields emitted by the human brain, and EEG measures the voltages on the head. Both are products of the direct products of the electrical current flowing through the neurons. So they are they're a direct index of brain activity. Specifically, here in MIT, we do have a MEG facility or magnetoencephalography, and you can see the device here. It comprised 306 sensors surrounding the head, and they measure the magnetic fields emitted from the human brain, which we can then use to reconstruct brain activity and estimate functional alterations due, for example, to Alzheimer's disease. The device is enclosed inside a shielded room to protect the measurements. It's not an affordable, it's not an ambulatory setting apparently, but later on I will explain what I mean. But it's kind of, it can be considered a gold standard for non-invasive electrophysiology. And you can see, for example, the instrumentation involved, the helmet structure, and we have different types of sensors, but all of them are sensitive to magnetic fields in the brain. And to get an idea on how powerful this method is to non-invasively capture brain function, you see here responses to binaural stimulation using auditory beeps. By averaging some trials, we see the evoked responses, and you can see here a deflection 100 milliseconds from the stimulus onset, localized in the right and left auditory cortices. So it can be it can have very high resolution, and importantly, it captures all the dynamics involved. Also here, I can show you the dynamics involved when we present a human face to a participant. Shortly after the presentation of the stimulus, you will see early visual cortex activity, which then propagates rapidly in more anterior sides. And eventually you see hotspots of activity in the parietal cortex, where the human brain has dedicated areas to process categorical stimuli like faces, objects, scenes. So all of these dynamics can be captured non-invasively, and we can use them to study brain activation. And very briefly, to finish with that, you can see how MEG is a powerful method. Yes?
If you take many participants and show them the same face, would you see exactly the same pattern?
That is a good question. If we would see the same patterns across participants, if we were to repeat the experiment. And the answer is largely yes, in a sense that, at least in this kind of stimulus I presented, we know very well, I mean, we don't know very well, but we know a lot about the activity involved and the computations in the brain. And actually, after light hits the retina, information through the thalamus, lateral genicular nucleus, goes posterior to the early visual cortex. So in all participants, we will see early visual cortex activity. And then within about 130 to 170 milliseconds, we will see the area, like for example, fusiform gyrus, which has to do with face processing, activated more or less in all participants, because that's how we process faces. So this is very consistent across participants. Of course, there are components that vary across participants as well. That was a good question. And basically with this kind of signals, we can see what participants see, perceive, remember using decoding techniques. So we can actually use machine learning. We can read what the brain encodes, what kind of information is encoded in the brain. And here, for example, I saw, like when we present different images, we can see how the brain encodes information about gender, age, identity, or familiarity of the face in front of us. And we saw, like in this case here, that gender and age information is encoded faster than identity information. So we can capture and describe a lot of these dynamics and information encoded in the human brain. Also, we can answer questions like what are the dynamics that the brain encodes information about objects invariant to position or size. We see, for example, here that size invariance is achieved before position invariance in our human visual system. We can also decode information about auditory sources. We can also decode information about more complex cognitive information like social dynamics. And that's meant to show you that non-invasively, we can actually capture a lot of information about what is happening in the brain. And so let's use this for this project here about Alzheimer's disease. And that was actually, and I'm very thankful for this, a project that was originally funded by J Clinic and allowed us to proceed with this work. So that's about developing an embedding method, graph embedding method, and use this to predict Alzheimer's disease in patients. So specifically, the project involved the analysis of 76 mild cognitive impairment patients, of which 48 were stable and 28 were progressive. What I mean by that, following the population 3 years after the data collection, 48 of them still satisfied the criteria for the MCI inclusion, whereas 28 of them progressed to Alzheimer's disease. And we also had a control population of 53 aged matched clinically normal subjects. All of this data came from the Madrid cohort. They have a very similar MEG device in Madrid as well. And that's from data from Fernando Maestre, my collaborator in Madrid. The data comprised 5 minutes of resting state data. So participants sit in the MEG and they don't do a task. And we record data from 306 channels in the MEG device. The demographics of this population were matched between controls and MCI patients, mild cognitive impairment, with only a small exception, possibly the gender, which tended to differ, but I don't think that was a major issue in this study. To proceed with this, to work on this, ourselves and also a lot of people in the community were influenced by the seminal study by Brack and Brack in '91. And they actually showed the neuropathological staging of Alzheimer's disease, and that it follows characteristic paths. It's not random. These proteins accumulate in characteristic pathways. For example, the amyloid deposits first accumulate in basal portions of the cortex and then spread to association areas and then all over the cortex, having toxic effects. Whereas tau proved to be more easy to stage the disease. Initially, we have tau accumulation in transentorhinal region, then spreading to entorhinal regions, and then all over the cortex, causing destruction. That was a study, an autopsy study of 83 brains of individuals with dementia 30 years ago. And motivated by this, it's reasonable to expect that since characteristic areas are involved in the progression of the disease, we will have characteristic functional alterations in brain areas as well, characterizing the staging of the disease. So in our work, we focus on functional brain networks, meaning we quantify how one brain area communicates with another brain area and having this ROI by ROI connectivity. I am going there, how to measure this connectivity. And actually, thank you for the question, because that's actually answered in this slide here. So starting with the measurements in the sensor level, we use algorithms such as minimum norm or beamformers or some other variants that allow us to reconstruct activity on the cortical surface. So no, in this case, now I went to our study. So non-invasive, in vivo, measuring of brain activity and reconstructing functional brain networks. Sorry for the confusion. The motivation before was that we already know 30 years ago that these proteins accumulate in characteristic paths. So it's reasonable to expect that some sort of functional alterations, connectivity in the brain will happen also in these paths, rather than random varying by Bayesian. So there must be something common, and this common should be captured also non-invasively by our functional measurements. And this is how we construct these functional measurements now, these networks of how the brain communicates all around. So we produce activation maps using our measurements, and then we use different metrics to establish cortical interaction. It can be coherence, for example, which is the generalization of correlation in the frequency domains. It can be phase synchrony, which we often use, showing how different brain regions align in phase. Or it could be more complex measures, such as Granger causality, that shows how one brain region predicts the time course of another brain region. There are many, many tools. That's all signal processing tools to use, ultimately to construct this kind of brain network. So one brain area is related to another brain area. And this means that we actually move to the domain of graphs, brain networks, having nodes, which are brain regions, and how they interact with other brain regions through edges. So we're no longer in the Euclidean space, but rather in non-Euclidean spaces. And this takes us to geometric or graph deep learning. And that's a field that is actually exploding. You may know that as well with all your studies here in CSAIL. So we have, other than not only human brain networks, but of course we have financial networks, protein networks, social networks. Our world is full of data best represented in networks. And there are specialized tools that are being developed for this now. One of them is graph neural networks that generalize the operations of the neural networks to the graph domain. For example, convolution, which is easy to describe in a Cartesian grid. But when you go to non-Cartesian grids, then it can be more complicated. But there are several tools for that. For example, convolution is multiplication in the spectral domain. So there we have ways to take the data in the spectral domain, and then we apply multiplications in the graph neural networks. And there are also pooling operations. These tools, though, are mostly used in a supervised way, and we wanted something unsupervised that would be more robust, having less data than the vast amounts of data typically needed in deep learning. So we followed an approach that has to do with graph embedding, where we start from the network matrices. These are also called adjacency matrices. And then we, through some model, some encoder, we can map the data in a low dimensional space. And the data in this space can easily then be used for further tasks, downstream tasks, such as node importance, community detection, and so on and so forth. And the idea is that the structural properties of the graph, like the first or second order proximity, is preserved in the embedding space. And also node similarity in the high dimensional space can also be obtained by computing the distance in the embedding space. So more specifically, we extended a model graph to Gauss to multiple graph to Gauss. And the idea is that it gets multiple brain networks and then has an encoder, which is basically fully connected layers to construct hidden representations that are shared up to this stage, but then they are split for the mean and variance of Gaussian distribution characterizing every node. So we achieve what is called Gaussian embeddings, or intuitively, every node in the brain network is described as a Gaussian distribution with the mean and the variance. And this has the advantage that we can also model uncertainty in this embedding space. And I have a question here, yes?
Yeah, what objective do you use for training the
So that's a good question. So I think the previous speakers in this lecture series talked about contrastive learning, and I will do the same here. Intuitively, the loss here has to do with contrastive loss. Having positive and negative samples. Positive are the ones that are nearby nodes in the original space, like 1 or 2 hops. Whereas the negative pairs are nodes that are far, like 3 or 4 hops in the network. So by optimizing such a criteria, you really force the first, second, third order structure of the network in the embedding space as well.
I was wondering your encode graphs. Did you register the brains first? Did you enter the whole?
If you can speak a bit louder, I'm sorry, I couldn't hear the question.
Register the brains first because my knowledge is the 5 before.
If I did, what do the network then? Oh, register. Oh, that is a very good question. And actually, I will answer this if I go to the next slide, and you will see why if I register the brain networks before. And the answer is, okay, let me then answer this by yes, they're registered because we are applying these networks in regions of interest, which are basically corresponding to from one brain to another one. So yes, we register the brains first before doing the analysis. They're in the same original space as well.
So it's a follow-up question on Adam's about what objective function you use. So this constructive bus helps drive the new ticket.
Yes.
What about the variance? Like, how do you learn?
Yeah, so the distance is quantified using the Wasserhoff distance, which is basically probability distance. So it takes into account both mean and variance. So the distance is using both mean and variance. It's a probabilistic in a probabilistic setting. That is a good question. Okay, so what we really did step by step in this work is we started with the MEG resting state data. We used an atlas to basically define regions of interest and then summarize activity in every region of interest with a single time course, activation time course, in 68 regions of interest. And then we used, in this case, actually mutual information to construct a 68 by 68 matrix of how one brain region is coupled with another brain region. And this gives us the brain maps. And then we used this Gaussian embedding method to embed the graphs into a lower dimensional space where we have the mean and variance for every node. And using this mean and variance in every node, we then use some more classical machine learning tools to, at this stage, do some analysis like classification across different groups or assess effects in different brain areas. What I mentioned before is that, in this embedding space, we map the data in this lower dimensional embedding space. What should this low dimension be? We use the surrogate task, link prediction task, which basically we had the brain network predict whether an edge is actually missing or not in test and validated data. And we found that the optimal dimension to represent the embeddings is 16 in this case, both in the validation and the test set. So I used the 16-dimensional embedding space. And with this embedding, like I said, we used more classical tools. We actually used the average of 10 traditional classifiers, such as random forest, SVM, linear SVM, or radial basis SVM, or other tools from scikit-learn to average and have a more objective performance of to quantify how much information is in this embedding space, and compared it with another technique for embedding, Node2Vec, and this one doesn't have this uncertainty quantification in the embeddings as well. It's a more traditional approach. And we saw that in all cases tested, the graph-to-Gauss model did have a superior performance, and on average, across all these classifiers, we achieved this performance discriminating different groups. So from neurotypical controls, versus stable MCI, we had 79%, and roughly this performance in the other comparisons. We also used some statistical tools in using resampling and beyond, to describe beyond the context of this talk to describe here, but we quantified which brain regions were involved in discriminating groups, and we found frontal and parietal sides consistent with other literature using other modalities to find effects. And one of the areas was the parahippocampus, which is involved in memory formation, so directly relating to Alzheimer's disease. I think I have a question around here.
Yeah. So if I understand your graph correctly, you're doing certain types of classification based on the embedding.
Correct, yes.
So, and you say you use like an SVM or something like that with a
Among others, yes,
yeah,
10.
if you have these probabilistic embeddings, so like mean invariance, which represent the distribution of possible features for a particular node, like what do you give to your classifier, which would expect, say, the input vector, like do you marginalize possible representations? Or?
I believe we used all the features, the mean and variance as they were. I don't think
Okay, so you just
yeah. Unfortunately, Mungji, the postdoc that worked on this, had an emergency meeting somewhere else. He would be here to answer more specifically, but she's not here to respond to that. But I believe that was what she did. Okay, and so with these and others as supporting work, we actually were able to fund this work and proceed with a large-scale project now to create a low-cost ambulatory test using these and other tools. So specifically, we now aim to make more powerful, more robust biomarkers by incorporating heterogeneous data, not just these functional networks, but also epileptiform activity. And epileptiform activity are basically paroxysmal sharp waveforms, or in simple words, spikes, present on MEG or EEG data, and that they are very transient and disrupt background activity. So I'll motivate further in the next slides. And another aim is to convert this to a more low-cost setting using electroencephalography rather than the MEG device that is a more research tool. And EEG, maybe you're all familiar with this, but basically uses cups like a swimming cup, but with electrodes attached to measure voltages on the scalp, for which we can also use to reconstruct brain activity. And by mentioning heterogeneous data, you may start thinking about multimodal learning, as a hint here. So specifically, for Alzheimer's disease, I mentioned already the accumulation of proteins, amyloid beta and tau, which cause functional changes, like, for example, here, hippocampal hyperactivation in the stage of mild cognitive impairment. But the community has also noticed, even from the earliest cases of Alzheimer's disease, that they also have epileptic activity oftentimes. So there is some comorbidity here between seizures and Alzheimer's disease. So patients with Alzheimer's disease have a 10-fold higher likelihood of having seizures than a general population, and lower scores in neuropsychological tests if they have seizures. So the cognitive decline is also stronger there. But it also goes the other way around. So patients with seizures have increased risk of developing dementia and also increased amyloid beta burden. So it seems that there is a very close relationship between these. And if you ask me, about the mechanisms, how this is happening, it's extremely complicated, like almost everything involved in this neurodegeneration disease. There are tons of possible neurotransmitters and pathways involved. One general, hypothesis is that the brain has complex networks of excitatory and inhibitory neurons. And the toxic effects of amyloid oligomers may be affecting primarily inhibitory neurons. So by being toxic to inhibitory neurons, then we have hyperactivity in the brain, and this hyperactivity in the brain is causing also epileptogenic activity. These spikes in the neurons. But the mechanisms are super complicated and definitely beyond the scope of this talk here. I'm by far not an expert for this as well. But in terms of using this, it's quite well known that this relationship exists, and one can use it to predict the onset of Alzheimer's disease. So in the cohort of 63 amnestic mild cognitive impairment patients from the Madrid cohort, having these MEG recordings, we found, our team went through all these recordings and found in 31.75% of these patients, we had epileptogenic activity. And so we split the population into two groups, with and without and with epileptogenic activity, and we also found some neural effects. This brain region here had reduced coupling in the gamma frequency, like neural oscillations of 30 hertz and above, had reduced coupling with two other brain areas. One is in the frontal cortex, the other is the precuneus, which has a lot of functions. One of them is also related to memory formation. So there is definitely a relation between epileptogenic activity and mild cognitive impairment and some neural effects. To show you examples of the epileptogenic activity that we identified, you can see here in the left temporal cortex, which is really often the site for seizures in epileptic patients. We found an epileptogenic spike, epileptiform spike, I think, to be more accurate. You can see here it is captured by the time course of many sensors. The most pronounced one is shown here, and that would be the overall topography measured outside the head of the magnetic fields, and this shows the localization of the spike in the temporal cortex. And another example from another patient, we can see this time in the right frontal cortex. But what we know from literature and what we've seen in our data as well is that the location is not really very informative of the Alzheimer's disease staging. At least that's what it is believed. Okay, so with this in mind, we also want ambulatory measurements, like I said, and for this, the technology is actually improving rapidly, and now have dry EEG systems, which are very easy to set up. You can see here with high and low electrodes. They're easy to set up, applied without much assistance, because they have these multi-pin electrode designs that go through hair and establish a good contact. And to prepare them, it doesn't really matter how many electrodes you have, more or less, it is easy to set up. And also here shows comparisons between wet and dry EEG, showing very good correspondence. So that's actually the instrument we want to use. It can be used in home or in residential settings. But I also want to include for completeness that in the future, maybe not so far, we can also be having ambulatory MEG, because we have new types of sensors now called optically pumped magnetometers, which are the new exciting technology for MEG. We can move away from these large bulky devices that we cannot move to more theoretically portable, wearable, and having a better signal. But right now, these are all theories and there are prototypes, but the community is moving really fast. So maybe if I am to give this talk a year from now or two, I may be telling you different news as well. Now there are several prototypes actually of these devices. So there is certainly a possibility for this to become a reality in the near future. And so in our research design, we will be recruiting 200 amnesic mild cognitive impaired patients in a span of 5 years. So this will take a while. And they will be recruited in the University of Texas Health Science campus. And of this, we anticipate 70% conversion rate yearly. from AMCI to Alzheimer's disease. That's reported in literature. And we will be collecting several recordings from them, MEG and EEG, which are considered a gold standard. That would be a wet EEG in this case. But we will also be collecting ambulatory EEG with the dry electrode EEG system we already have, comprising 256 actually channels. But the goal here is to see if we subsample them, how many do we need? And if 32 or 16 is enough actually to make inferences as well. So we can go that way too. We will be capturing epileptiform activity and characterizing it. And we will also be constructing these brain networks, as I said. And using this data, we actually now aim to use multimodal learning using MEG and EEG networks, using the multiple graph-to-Gaussian encoder, like I mentioned before, jointly embedding them in a common space using contrastive loss, and that would be same or different patient here. And after do this multimodal learning, it will allow us to quantify unimodal or supervised predictions. Like, for example, Paulina Goland mentioned some lectures back, only in her case was text and image embedding, but we can do it in these modalities. And then eventually, and I'm almost done, eventually using this encoder properly trained, we can have an ensemble model that takes into account the epileptogenic activity and age, gender, and have an aggregate score. So unfortunately, I don't have any results for this, but that's actually our aim. It's going to be a 5-year project. And we can also extend this to more complex learning because we will have MEG, EEG, and ambulatory EEG. So we can have a more complex, learning in a joint embedding space. But ultimately, the goal is to have, with a simple EEG scan, we could analyze the data that will be collected in home or in a residential facility and be able to make a prediction. And also in the same study, how epileptiform activity and aberrant functional networks relate to the disease. So thank you so much. I hope I didn't run out of time. I don't anticipate much in the panel, discussion since there's no clinical collaborator here. If you have any questions.
Tell us a bit more about the cohort that you used to evaluate the first part of your talk on the biomarkers with this Gaussian-based graph model.
The question is to say a bit more about the model that I used. Oh, about the cohort. Yeah. Okay.
Criteria. Tell us a bit more about the evaluation.
That would be a good question. The inclusion criteria are actually described in the article, which I would refer to you for more detailed information. It was not based on amyloid, beta or tau measurements with PET. It was based more on diagnostic clinical neurophysiological tests. I don't remember the exact inclusion and exclusion criteria though, but they're documented in the article. They were all mild cognitive impaired patients.
I guess what I'm trying to get at, what I'm trying to understand better here is, it seems a lot is still unknown about the natural progression of Alzheimer's and how it presents. It's not clear to me if one were to use a supervised approach to both refine and evaluate the biomarker, what the correct ground truth is. I'll also be comment on that.
Yeah, I guess the question has to do with some uncertainty in characterizing the disease staging and whether we can. I'm paraphrasing, but I'm trying to get the point. Whether we can characterize them reliably to be used in a supervised setting and all this. And my understanding is that this can be done. It differs even in hospitals and so on, like how clinically you would define one to be mild cognitive impaired or not, and it varies a lot. I do rely on my team for that. It's something that I didn't do on my own, so I find it hard to describe the exact parameters used in this case. They collaborated with hospital there, so they have done that in some objective way, I hope.
Correlation with outcomes. So outcomes meaning, for example, time to death or time to particular disability.
So because they monitored them in 3 years, we know that many of them went to the Alzheimer's disease, converted eventually, and I think they're still monitoring them. It's not very uncertain actually at this stage of the disease, even though clinically it's hard to define them. I think the impairment is very clear and it's much more well defined than in early stage, which is the subjective cognitive decline, which is basically someone reports, I don't remember well, or relatives say that he attempts forgetting. That's before. And the mild cognitive impairment, there are scores that show this, so it's more well defined cohort of patients.
Yeah, so I had a question. So I'm not too familiar with brains, but it seems that you went from like a high resolution 3D video to like these discretized graphs. Are you losing information by making this simplification, or what's the benefit of like already on this graph instead of the continuous?
That's a good question. It has to do with moving, transforming our data from the original sensor space or the more extensive sensor space that, source space that we have brain activation maps to the representation of graphs where we represent relationships in brain, across brain regions. And one could consider that some information may be lost, but to some extent we need to do that to align subjects, and also keep in mind that the information may look quite rich and extensive, but it's not necessarily that much because it comes from, in the MEC case, 306 sensors, but the actual signal space is much smaller. If you are to do PCA, and see the dimension is much smaller. So the information that is there is much smaller than this very high dimensional space in the voxel level to begin with. And these are large scale networks. So basically with macroscopic measurements, we cannot have the resolution to discriminate voxel to voxel, even though we reconstruct using heavily regularized maps. So by capturing the large scale relationships, I think more or less we are capturing the true underlying information that is there without wanting to exclude other possible things. In fact, there are many studies using this data for the same purpose, to predict Alzheimer's disease, showing other measures such as power spectra that are changing, but that's not very specific. Like we often have a slowing of the brain when we have dementia or schizophrenia or other diseases. So that's a very general biomarker, the slowing of the brain that you can see with the time courses and the oscillations. Or there are measures of complexity of the neural data as well, the time courses. But we think, and we're not alone in the community, that the brain networks have more information to specifically identify and characterize diseases like Alzheimer's disease.
So thank you very much. And yeah, I'm sure Dimitrios can answer some question afterwards if you have a question. So